In this module, I will discuss about extensions of Mendelian concept, incomplete dominance and multiple alleles. What is incomplete dominance? When the heterozygous phenotype is midway between the phenotypes of the two homozygotes, the partially dominant allele is called incomplete dominance or partially dominant. In this example of incomplete dominance in Snapdragon, we have crossed a red colored flower with a white colored flower with genotype of capital WW and small ww. In the F1 generation, we obtained a pink colored flower, capital W and small w. When they are self-fertilized in the F2 generation, we obtained one red, two pink and one white colored flower with a phenotypic and genotypic ratio of 1:2:1. So here there is a one red colored flower, one white and two pink colored, which is a case of incomplete dominance. So here the allele for red colored, that is capital W, is incompletely dominant over the allele for the white colored small w. Therefore, WW, capital W, small w have pink color. So what is the genetic basis of flower color in Snapdragon? Now phenotype, that is red, which has a genotype of capital WW, amount of gene product, that is pigment production, is twice X. On the other hand, in the pink colored flower, which has a genotype of capital W and small w, here gene product is only X. On the other hand, in the white colored flower, that is small ww, amount of gene product is 0. So X, it is the intermediate between 2X and 0, so it become pink colored pigments, and pink colored pigments are produced over here. So this is the genetic basis of flower color in Snapdragon and an important example of incomplete dominance. Next comes multiple alleles. What is multiple alleles? In Mendelian concept, genes exist in no more than two allelic states. There is only two alleles are present. It was later discovered that gene exists in more than two forms, that is 3, 4 or more alleles. When a gene occurs in more than two allelic forms is called multiple alleles. One of the important example of multiple allele is coat color in rabbits. Here, color determining gene is represented by capital C. It has 4 alleles, that is albino represented by C, then Himalayan, it is represented by C and superscript H. On the other hand, chinchilla is represented by C and superscript CH while wild type is represented by C plus. Now, this coat color in rabbits, that is albino, which is represented by the genotype CC, and phenotype, that is white hairs over the entire body. So, it is called albino. On the other hand, in case of Himalayan, which is represented by a genotype of CHCH, it has black hairs on extremities. On the other hand, in case of chinchilla, which is represented by the genotype, that is CH and CH, that is here white hair with black tips. On the other hand, wild type coat color, which is represented by the genotype C plus, has colored hairs. So, here this is the case of coat colors in rabbits. Now, here the coat color gene has 4 alleles. Now, the dominance relationship of this coat color gene is termed as allelic series. Now, this dominant relationship among the alleles are as follows. The wild type allele, that is C plus, is completely dominant over all other alleles of the series, that is CH, then CCH and C. On the other hand, chinchilla, that is CCH, is partially dominant over Himalayan and albino. On the other hand, chinchilla and Himalayan are partially functional or hypomorphic. Now, albino allele C is non-functional. It is also termed as null allele. Now, there is another important example of multiple allele is human blood types. Human blood types, that is A, AB, B and O, and gene responsible for producing these A and B antigens is denoted by the letter capital I. So, capital I has 3 alleles. One is IA, one is IB and one is small I. So, I designation stands for isoagglutinogen, which is another term for antigen. Now, genetics of ABO blood typing. Now, A blood type, which produce only A antigen, and it is represented by the genotype IA or capital IA small I. On the other hand, the blood type B, which produce B antigen and is represented by the genotype IB or IB small I. In case of AB blood type, it produces both A antigen and B antigen and is represented by the genotype IA and IB. In case of O blood type, it produces neither A antigen nor B antigen and it is represented by small II. So, this is about the genetics of ABO blood typing. Now, in this case, the IA and IB alleles are termed as co-dominant, that is, both of them are expressed simultaneously and neither of them are dominant or recessive. So, they are termed as co-dominant. Small I allele is recessive to both IA and IB alleles. Now, I gene is also termed as polymorphic, which means it has many forms. So, this is the one of the also important example of multiple alleles, that is blood groups in human. Now, the biologist, that is English biologist Bateson, who has made a major contribution in understanding the Mendelian concepts, and he has made a lot of contributions in the science of genetics. In this module, we have provided key concepts of the module, quizzes, list of books for reading, and also some study materials. Thank you for attending our online course.